Today I learned about brain, human brain and animal brain. So first of all, in human brain, the cortex is much larger than the animals. It will overlap the cerebellum. So cerebellum is not visible in human brain. Second, there is a difference between rat brain. Rat is much bigger, but mouse is a still younger version as differed structurally. But the baseline of the all circulatory system of baseline circulatory system is same in all humans and as well as animals. So okay, so what was my question? My first question is that is there any difference between the ambitions center or motivating center which is present in humans as from zero achievers to high achievers, and the motivating or ambition center in animals? So no, there is no as a broad distinction between the human brain, motivating center, or any animal, because motivation is the basic skill of survival. Ambition to eat, to find good grass, in example both, and rat may have finding good breeding places. So yeah, they have same ambition center as we have. There will be difference in neurotransmitter stimulant. For example, some of us having dopamine hit watching someone doing something like climbing, but some of us will be stimulated have dopamine hit when while we climb ourselves. Second thing, uh, gray matter, white matter is the thing that carries all your nerve fibers and the gray matter contains all the cell glia, uh, which generate the signal. Okay, and there is a large neuron which is known as the internal capsule, which is 1 meter long, which carries all your data from the basal ganglia, which carries all your ambitions and motivations. Your dopamine goes into the spinal cord. The difference between the structure of human brain neurons may be more or less in some, but the basic structure is same. The human brain is folded for two, because our skull is not expanded to compress the surface area of the brain. From evolution, we have more complex brain system than the animals. To basically, different species have different evolved uh, brain to adapt in their specific environment. For example, the things rat can do, we can't do. For example, their olfactory bulb is much more developed because they are uh, dependent on the smell, but we are bipedal. We walk on the two limbs. We walk from two limbs, so we have less developed olfactory system. We have more developed eyesight system, that we are dependent more on the vision. Okay, and basic structure is same in all humans. For example, in high achiever or in low achiever, there is not much difference as we see, but there will be minor difference in in the neurotransmitter release, dopamine, and you know uh, signaling speed, signaling transmission, uh, capability of one neuron to contact with the other neuron. These things may vary, but the structure is similar. Okay, then what we can do from the dead brain to relate with it or to interpret about the live brain. So basically from the dead brain we can study neurons, which is the basic structure and functionality of the brain, and we can study about the anatomy of the brain, but we can't study DNA and molecular processes like RNA expressions. We can't study from the dead brain. We have to do it in the live brain. Brain functions or any kind of behavior we can't interpret from the dead brain. So today I learned this. Thank you so much.